review the DNA replication in eukaryotes. It takes place in the nucleus during the S phase of interphase. Let's start by looking at the enzymes that play a role during DNA replication in eukaryotes. Okay, these are the enzymes that play a role in DNA replication. The enzyme helicase breaks down the hydrogen bonds between the two DNA strands in the DNA double helix. Primase enters RNA primer. Topoisomerase untwists the double helix and takes out tension. DNA polymerase III joins 3 nucleotides into a new strand of DNA, starting at the primer. DNA polymerase I replaces the RNA primers with DNA, and DNA ligase joins the DNA segments on the discontinuous strand. Now there are a few other terms that you need to know. 5' and 3' end. So I want you to kind of always identify the 5' end and the 3' end. And then I want you to identify the direction in which the polymerases go, so any type of direction. And then I want you to identify the Okazaki fragments. Remember those?
Yes.
And I want you to identify the primer. You have the primase, the enzyme that will make the primer. Do you know what the primers are, or what are the primers, RNA or DNA?
RNA.
RNA primers, that's important. Another thing that's important to know is that the polymerase will read 3' to 5', but will assemble 5' to 3'. The enzyme helicase opens the DNA double strand. Then the topoisomerase removes any tension that is in the DNA double helix. The RNA polymerase or primase starts making RNA primers. The DNA polymerase III attaches to the primer and copies the complementary DNA strands. Now, what you see here, this is a DNA double helix, and I put some of the structure in there. It's not complete, but it's just like so that we get an idea. So how do we know what is the 3' end and what the 5' end is?
The leading carbon.
Yeah.
So 5 is.
Yeah, we have 5 here. So we have carbon 1, 2, 3, 4,
and
5. So this is the 5' end. And then here that would be the 3' end, so I draw the 3' end down here. And then of course on the other side we have 5' end, and then this here would be the 3' end. Okay. So the first enzyme that comes is the helicase. So what the helicase does, it will unzip the hydrogen bonds up here. So the helicase cuts the hydrogen bonds and makes the double strand partially into single strands. Then we have some proteins that actually will kind of stabilize the DNA. We're not going into that. Then we have the topoisomerase that actually takes the twist out of the strand. When you look at the models of the DNA double helix, it's kind of this twisted. So when we start cutting, we have some tension in there, and the topoisomerase takes the tension out. So takes the kind of cuts and closes it together, so takes the tension out. Now the DNA is ready to be replicated. Replication usually starts at what we call the point of origin, and we assume that the point of origin is just here, and the DNA is now ready to be replicated. The DNA polymerase 3 will do most of the replication, so copying the DNA, but it needs a primer. And it cannot make a primer, and there is no DNA polymerase that can actually make a primer. So we'll have an RNA primer, and so the RNA polymerase will make a primer. Let's say this here is my sequence, so that would be, let's say we have an adenine, a guanine, a cytosine, and a thymine in this DNA strand. So what would be the complementary primer in this case? If I have an RNA primer. So if my primase now comes and makes the primer.
It would be
So
uracil.
A binds to
uracil.
uracil, so we'll have a U. G will bind to
Cytosine.
cytosine,
Guanine.
guanine,
And then adenine.
thymine to adenine. So there is no thymine in RNA, so we'll have here this primer. The primer will be longer, but for our purpose, that's sufficient. Now the DNA polymerase will come and actually read the strand. DNA polymerase reads from the 3' end to the 5' end, but makes the DNA 5' to 3'. So we'll start here and add our big DNA polymerase. So that is now my DNA polymerase that kind of starts here with the primer, attaches to the primer, and then starts adding on molecules or bases. So let's say we have here another A, and then a G, and then the DNA polymerase will add, A will bind again here now with T, and then if it moves up here, it will bind the G and add a C to that. And between the DNA and its complementary strand, again we have the hydrogen bonds. So we'll have 2 hydrogen bonds here, 3 there, another 3 here, and 3 there. So the pol 3 moves in that direction, so it reads 3' to 5', but writes 5' to 3'. So what we see here, that would then be the 5' end, and it moves on. Now on the other side of the strand, it's a little bit more complicated because the polymerase can only write the 5' to the 3'. So once we have an attachment, or like we have access to the opening strand, the primase will make a primer here. So this is my primase. It will make my RNA primer. So this is the RNA primer. And then the polymerase 3 will kind of add the strand complementary. So this here is now the 5' end, and the 3' end goes in that direction. Now let's kind of do a mental step forward. Imagine this is the new part, or normally it would start here with a new part like that we had. Let's go back in time and look what happened before the RNA polymerase was able to just add a new primer here. At one point, it started adding a primer here, and then the polymerase would add the DNA. And then at the very beginning, when the whole replication process started, the first RNA primer was added close to the point of origin and then went to the end. So what we see now is that we have different we have those primers here, and these are all RNA primers. They now have to be taken out. And that is the next enzyme, that is the DNA polymerase 1. So let's say the DNA polymerase 1 cuts the primer, and exchanges it to makes it a DNA. So it takes the primer out. Actually, should I use green here? So it cuts that part out, replaces it with DNA. Here that should also be our second strand. This happened here again, cuts the RNA out, replaces it with DNA. But what we see then is that we have those little holes here. So there is a missing connection between those fragments. And those fragments is what we call the Okazaki fragments. And those Okazaki fragments are special to what we call, how we call this strand here. It has like two different names.
Leading.
That would be the leading strand.
Lagging.
So here we have the leading, and that would be the lagging strand, or the discontinuous strand. Discontinuous strand. Now, what the ligase now does, it will replace the hole, or kind of fill the hole, and connect this, make this bond. So we have the ligase putting the Okazaki fragments together, and that's kind of it. So we started with one double-stranded piece of DNA, and now we have two double-stranded pieces of DNA. Do you know when this DNA replication happens? We're talking about eukaryotes here. Do you remember what we did last week?
Mm-hmm.
Mitosis.
Mm-hmm.
And mitosis is just part of the cell cycle.
Oh, this happens during interphase.
During interphase?
S phase, like.
The
Yes.
S phase, it happens during the S phase of interphase. So when you look at when DNA replication takes place, so if we say we have our cell cycle here, this would be our cell cycle, and then part of the cell cycle is mitosis. And there we have those different phases, like mitosis starts with prophase, and then we have metaphase, anaphase, and telophase, followed by cytokinesis. This can overlap. So telophase and cytokinesis can overlap. Which is not part of mitosis. So mitosis is just this part here. This here is all mitosis. And before mitosis, we have the interphase. And during interphase is set up into three parts. So we have G1, S, and then G2. And the S phase, that's the important one, where we have the DNA replication. During S phase of mitosis, not of mitosis, during S phase of the cell cycle, so that prepares or duplicates the chromosomes actually. So what the DNA replication is just duplication and getting a cell ready for mitosis, or actually also for meiosis. Great, thank you.